Hello everyone, today we start a new set of experiments about the enzymes, and in this set of experiments we will study the activity of cell-biase, which is the enzyme that digests cell-biose. For the first experiment, in which we are going to build a standard curve for p-nitrophenol, we need 5 spectrophotometric cuvettes, 5 15 ml tubes labeled S1 to S5, then the spectrophotometer set up to measure absorbance at 410 nanometers, the stop solution, DI water, and p-nitrophenol. We will also need the pipettes and pipette tips. Label 5 15 ml tubes S1 to S5 and add 1.25 ml of DI water into each of the tubes. Add 500 microliters of 1 millimolar p-nitrophenol, which is our standard, into tube labeled S5. Add 750 microliters of DI water to the same tube. As a result, you should have 2.5 ml in tube 5 and 1.25 ml of solution in the rest of the tubes. Take 125 ml from the S5 tube and add to the tube S4. In this activity, we are going to do the two-fold serial dilution. Now mix up solution in tube S4 by pipetting up and down several times. Repeat the procedure and take out 1.25 ml from the tube S4 into the S3 tube. Mix it well, and then do the same for the tube S2. Discard 125 ml of solution in tube S2 and do not add anything to the tube S1. Now we have all 5 tubes with 125 ml of p-nitrophenol. Since we have different concentration, what are the concentrations of p-nitrophenol in each tube? Add 125 ml of stop solution to all 5 tubes and mix. Observe change in color. Label your cuvettes S1 to S5, and then transfer 1 ml of S1 solution from the 15 ml tube to the spectrophotometric cuvette. S1 solution is water and stop buffer. It serves as a blank in our experiment. We are going to use it to calibrate our measurements. Repeat with the transfer of 1 ml of remaining tubes S2 to S5. Make sure you do not mess up on this stage, because the S1 to S5 cuvettes will give you the rising concentration of the product. Now we are ready for absorbance measurements, and the spectrophotometer is an instrument that allows us to measure absorbance. Place your sample into the spectrophotometer, close the lid, and blank the spectrophotometer by pressing on calibration button. This would mean that we set it to 0 for the sample number 1. Now measure the absorbance for remaining samples. S2, S3, S4, and finally the highest concentration S5. Here you could see the table of the results and the standard curve. You will need to write it down into your lab book.